Hi students, so today in this video you are going to learn about part second from reproduction in lower and higher plants. In this part second you are going to learn about the structure of anther, microsporogenesis, structure of ovule, Megasporogenesis. So let us start from first, that is structure of anther. You are well known about the male reproductive valve from flower, that is nothing but androecium, and individual member from the androecium is said to be the stamen. Within the stamen it shows the presence of filament, connective and anther. So first of all, structure of anther. Immature anther show the presence of parenchymatous tissue at the middle or at the center, and which is covered with a single layered outermost covering called as epidermis. Anther show either one lobe or two lobes. If it shows the presence of two lobes, as shown in diagram, this is the one lobe, this is the second lobe. So such anther is said to be dithecous, and in each lobe it shows presence of two pollen sacs, and hence total are nothing but 4 pollen sacs in dithecous anther, and hence it is said to be tetrasporangiate. Okay, dithecous tetrasporangiate anther. And if one, then it is said to be the monothecous and disporangiate anther. So in such anther, the cells are divided into innermost sporogenous cell and outer primary parietal cell. Okay, so they are togetherly said to be the archisporial cell. Now these inner sporogenous cell will give rise to the microspore tetrad. This microspore tetrad is also said to be the pollen grain. The another name of microspore is nothing but pollen grain. And why they are said to be tetrad? Because they are present in a group consist of 4 microspore in a single group. That's why they are called as tetrad. And outer parietal cells will undergo divisions to form anther wall layers, which covers pollen sacs, and these microspores are present within the pollen sacs. Now let us see in details about anther. Anther show the outermost covering called as epidermis, followed by endothecium, and endothecium is again followed by middle layers. So all these three covers each pollen sac including the tapetum. So actually the anther consists of wall layers and pollen sac. So like this, there are 4 pollen sac. This is one, this is second, this is third, and this is the 4th pollen sac. Like this, there are 4 pollen sacs in dithecous anther. Okay, so we will study detail about wall layers. First wall layer is nothing but epidermis. It is the outermost single layered structure in which the cells are flattened or tubular cells, and it carries the role of protection. They are protective in function. Now this epidermis is followed by the second layer called as the endothecium. It is a subepidermal layer. Subepidermal means present below to epidermis, and it consists of the radially elongated cells with fibrous thickenings. It is like this. It covers the whole anther. This one you can see. This is the anther. Okay. Third layer is middle layer. Middle layer consists of 1 to 2 layers, and it covers each pollen sac. Here you can see each pollen sac is covered by middle layers. Last one is tapetum. Tapetum is innermost wall of anther or innermost layer of anther, which is nutritive in function, which provides nutrition to developing microspores. That is, it encloses the MMC, that is microspore mother cells, which are present within the pollen sac here. So here large cells are nothing but large cells. You can see these are large cells said to be the tapetum, which covers each pollen sac. Each pollen sac is covered with tapetum. So let us see the next, that is nothing but microsporogenesis. What is meant by microsporogenesis? Microsporogenesis is nothing but process of formation of microspores, that is pollen grains, and it takes place in pollen sac. Okay, so how the microspores are produced, we are going to see, but before that we will see a structure of microspore. So what is pollen grain or what is microspore? Microspore or pollen grain is non-motile, haploid, unicellular and uninucleated structure. Means it can't move from one place to another place by itself. It consists of haploid cells, haploid number of chromosomes, and it is unicellular body, consists of single cell and again single nucleus. Okay, and it is covered by two-layer wall called as sporoderm. And what are those two layers? Outermost layer is called as exine and inner is called as intine. Okay, so this outermost exine, which is thick and made up of the complex non-biodegradable, you might know the word biodegradable, okay, substance called as sporopollenin. And in some pollen grains it may be smooth or with sculptured pattern. It depends on the characteristics of that species. It resistance to chemicals, and at some intervals you can see in this diagram, this is nothing but the exine. This one is exine. At some intervals it interrupts like this. Here one, this is second, this is the third. Interruption, or at some intervals it becomes thin and it forms germ pores, and these germ spores are responsible for emergence of pollen tube during pollen germination. So remember, exine, germ pores. Okay, now let us see the next wall or second layer of sporoderm. Second layer is nothing but intine, and this intine is made up of cellulose and pectin. In diagram you can observe intine. So this is nothing but intine. Okay. Due to the presence of outermost covering, there is the enzyme which are thick and resistant to chemical, pollen grains become viable. So what is meant by viability? Viability is nothing but the functional ability of pollen grain to germinate, to develop or to form male gametophyte. So it depends upon the environmental conditions like temperature, humidity, and in rice and wheat it is 30 minutes. The viability is 30 minutes, and in some members like Solanaceae, Rosaceae, Leguminosae, it lasts even up to months. So the next point is nothing but development of male gametophyte. How male gametophyte is produced? Let us see. Male gametophyte is produced from pollen grain itself. Okay. So first of all, these pollen grain will undergo one mitotic division, and again it is followed by second mitotic division. So there will be two mitotic divisions. So when there is first mitotic division of this pollen grain, you can find this is the whole pollen grain, and which undergoes first mitotic division, you will get this structure. Okay, this structure. So it forms two unequal cells. Larger one is said to be the vegetative cell. This one is the vegetative, which is larger one. This one is said to be the vegetative cell, which is bigger and naked. Another one which is formed is smaller and thin walled, called as generative cell. Okay. This generative cell is suspended within vegetative cell. Vegetative cell is rich in food, and it shows the presence of irregular nucleus. And in this cytoplasm of vegetative cell, generative cells floats. Okay. Now, this is the two-celled stage prior we have studied, and most of the pollination is taking place at a two-celled stage in angiosperm. Now the next mitotic division is taking place in generative cell. Vegetative cell remains as it is, and the second mitotic division is taking place in generative cell. Thus it forms, here you can find two, this is one and this is second, two cells which are said to be the male gametes, and here it is vegetative nucleus. Okay. Now, due to the absorption of sugary substances from stigma, pollen grain grows in size and the intine comes out. Here the intine comes out through germpore, forming pollen tube. This is called as pollen tube, which consists of two nuclei which are the result of division of generative cell or generative nucleus, said to be the male gametes, and one tube nucleus which is of vegetative cell. Such three-celled structure along with pollen tube or three nucleated structure along with pollen tube is called as male gametophyte. Okay, and this point already I have told you, pollen grains are shed from the anther at two-celled stage in angiosperms. Now let us see about what the things which are taking place in gynoecium. So you are well known, gynoecium is nothing but the female reproductive valve, and a single member is called as carpel or megasporophyll. So carpel consists of what are things present in carpel? It shows the presence of the basal part called as ovary, style, and the topmost part said to be the stigma. It is called as stigma. So let us see what are the types of carpel. In some plants, it shows the presence of free carpels. They are not joined with each other. Such flower is called as apocarpus. And in some, it is fused with each other, said to be the syncarpus. Examples are Michelia and brinjal respectively. So already I have told you, in carpel consists of three parts: ovary, style, and stigma. The number of ovules in the ovary depends on species to species. It varies from species to species. In paddy, wheat, and mango, it is uniovulate. Ovules are not more but single, that is said to be the uniovulate. And if the ovules are more, then it is said to be the multiovulate, which are present like in tomato and in lady's finger. Let us see the structure of ovule. There are three types of ovules. First one is nothing but you can say anatropous type of ovule, which is shown in diagram. Second type is nothing but amphitropous, and the third type is called as just a minute. Let me show first the differences: anatropous, amphitropous, and third one is called as orthotropous ovule. So what are the differences among them? First, in the definition of anatropous ovule, you will get an idea about anatropous. Micropyle is directed downward. Here you can see micropyle, which is directed downward, and it is near to the funicle. Such type of ovule is said to be the anatropous ovule. If the funicle and micropyle are opposite, then such ovule is called as orthotropous. This is the funicle, and if the micropyle is at this position, such ovule is called as orthotropous. And if micropyle is at 90 degree to funicle, then the ovule is said to be the amphitropous ovule. So these are the three types of ovule. Here we are going to study about anatropous ovule. Okay. So what are the parts of ovule? Let us see what are the parts of ovule. First one is nothing but the placenta. So what is placenta? Placenta is the tissue present in ovary on which the ovule arises. So this is the placenta on which ovule has arisen. Next part is funicle. What is funicle? Funicle is nothing but stalk of ovule which holds ovule or which is attached to the placenta. That stalk is called as funicle. Next part is hilum. Hilum is nothing but the place of attachment of ovule to funicle. It is called as hilum. Here you can see this is the placenta, this is the funicle, this is the hilum. Next, nucellus. The parenchymatous tissue which are present at the middle part of ovule here. Later on it becomes or it get converted into the embryo sac. Such part prior or in immature ovule, it is said to be the nucellus. So integuments. What are integuments? Integuments are nothing but the covering which covers the inner part or surrounds inner part, and the role is nothing but protection. Here you can see two integuments. This one is the outer and this one is the inner integument. Next is micropyle. What is micropyle? Micropyle is nothing but interruption in integuments which lead into the opening is called as micropyle. What is the role of micropyle? Micropyle is responsible for the entry of pollen tube in embryo sac. Next is chalaza. Chalaza is nothing but opposite structure to micropyle or opposite pole of micropyle. This part is called a chalaza. And embryo sac. Embryo sac is multicellular structure embedded within nucellus. Nucellus मध्ये असणारं structure जे आहे, that is said to be the embryo sac. This part. Now let us see how the embryo sac is developed. That is said to be the Megasporogenesis, process of formation of megaspores. That is nothing but ovule. Actually, ovules are said to be the megaspores. How they are developed. In the prior diagram you have studied, that is nothing but a mature ovule which consists of embryo sac. Okay, that process is called as Megasporogenesis, and it develops from megaspore mother cell. Okay, so let us see development of female gametophyte. Megaspore mother cells. Here, within the nucellus, some of the cells will work as archisporal cells, and out of archisporal cells, some will behave as megaspore mother cells, which are diploid in structure, and it will undergo meiotic division to give 4 daughter cells, which are now haploid. Due to meiotic division, the chromosome numbers reduced to half, and they are said to be the haploid megaspores. Out of 4 haploid megaspores, 1 will remain as it, and it will take part in further procedure, and 3 upper will degenerate, and such one remainder is called as the megaspores, or it is also said to be the embryo sac. So it will develop in embryo sac later on. So it starts growing in size, and it well nourished by nucellus, and it will undergo 3 mitotic divisions. So first mitotic divisions will lead into the formation of 2 cells or 2 nuclei inside. Now these 2 will undergo again the second mitosis, formation of the 4 cells. Now the third mitosis division will lead into the formation of 8 nuclei. 1, 2, 3, 4, 5, 6, 7 and 8. Out of 8, 3 will go towards chalazal end. This one is a chalazal end. 3 will go towards micropylar end, and these two come at center and form as a single cell at the center, and thus Embryo sac shows the presence of 7 cells, 1, 2, 3 at chalazal, 3 at micropylar and 2 cells which are going to form a single. That's why this is a 1 cell, counted as 1 cell, 3, 3, 6 and 1, 7 cells. But it shows presence of 8 nuclei, 3 at chalazar, 3 at micropylar end and 2 nuclei at in middle. Okay, so that's why here the Megasporogenesis is said to be the embryo sac is said to be the 7-celled 8-nucleated structure. Okay, so in angiosperm the development of female gametophyte is endosporic, which is taking place within the megaspore, and it is also said to be the monosporic, which is developed by a single megaspore. That's why it is called as monosporic. Okay, so you have studied how the female gametophyte is developed. Here the female gametophyte is colorless, endosporic and monosporic. Thanks.